Good morning. My name is Jacob Marlin. I'm the Executive Director of the Belize Foundation for Research and Environmental Education here in Belize. I'd like to first thank the Turtle Survival Alliance for hosting and putting together this great conference in this difficult time. It's a tremendous resource for people all over the world, so I'm very happy to be a part of it. This morning, I'm going to talk to you about the conservation actions leading up to and including the first rewilding or reintroduction of Dermatemys mawii, also called the Central American river turtle, or in Belize, it's called the Hicatee, in the country of Belize. But first, I want to give you a little bit of background on the species. The Central American river turtle is the lone surviving member of an ancient lineage of turtles dating back almost 65 million years. It is the only living species in the family Dermatemys, so that makes this one of the most endangered turtle families in the world. The geographic distribution of Dermatemys is relatively restricted. It's found in the coastal lowlands of southern Mexico, a small portion of northern Guatemala, and portions of Belize. Currently, it is listed by the IUCN as critically endangered, meaning it's nearing extinction. The cause of its decline throughout its range is almost exclusively for human over-harvesting for human consumption. It is traditionally a diet that's been used, particularly in Belize, but other countries as well. It's a highly prized meat, and so it's collected by free diving, by using nets, harpoons, in a variety of ways. I'd also mention that even though the species is critically endangered in Belize, the laws do allow for sustainable harvest, and there's a closed season. There's restricted areas that you cannot harvest Hicatee, but they do have laws that allow for only personal consumption. There's no commercial activities allowed. No selling or purchasing of the species. Very briefly, I'll try to run through these things that we do know about Hicatee. This is a very large, fully aquatic turtle. They can get up to almost 50 pounds. They have relatively long lifespans, at least 45 years or more. They inhabit deep freshwater rivers, lagoons, tributaries, and other water bodies. They appear to be completely herbivorous throughout their entire life. They are primarily nocturnal, though they are somewhat active during the day. This is a species that does not bask. You will never see one of these turtles basking on a log or on the bank, never. They tend to bury themselves in the bottom of these water bodies, often in debris or in cut banks, leaf packs, relatively hard to find. As adults, they are sexually dimorphic by color, which I'll show you a picture of in a minute. They obtain their oxygen both from regular through their nasal cavity, but also they have a gill-like structure in their gular area that allows them to extract oxygen underwater, so they can stay underwater for very long periods of time. They also nest below the surface of the water, which is very unusual, which I'll get into a little bit later. And these shallow nests are subject to all types of predation and natural disturbance since they're right on the riverbanks. They lay usually 1 or 2 clutches of eggs per year. Mean clutch size is around 11 eggs or so. And these eggs undergo an embryonic diapause or delayed development. And their incubation periods can be up to 300 days. The temperature affects their sex, so TSD, they have temperature sex determination. And we are beginning to understand that sexual maturity takes about 15 to 17 years to reach sexual maturity. The easiest way to determine the different sexes is when they're adults. On the left, you have an adult male. On the right, an adult female. When they're smaller, they really look almost identical. Today, I'm speaking to you from Belize Foundation for Research and Mountain Education, BFREE, in southern Belize, the Maya Mountains. And 10 years ago, TSA, Rick Hudson, contacted us down here in Belize to determine what we knew about the status of the species and sort of explore the conservation opportunities. They wanted to get involved in Dermatemys conservation in the region. So Rick Hudson came down along with some others, and after a series of meetings, meeting key stakeholders, government officials, a countrywide survey was conducted in 2010, and then a national workshop took place, and real momentum started to unfold in this country. As part of the partnership, or as part of the conservation actions, we determined that there would be a benefit of having a captive component for the program, to both for a number of reasons. One, to create an assurance colony as a hedge against extinction, and also to investigate the feasibility of captive breeding for a potential Head Start release and reintroduction program, as well, and most also very importantly, to conduct research on the species reproduction, reproductive biology, and ecology. Not that much has been done. There's been some great work done in Mexico primarily, but not much work has been done at that time. So, we, 2011, TSA and BFREE began a collaboration to create the Hicatee Conservation Research Center to undertake these activities. And 10 years later, what we have is a facility that has 2 large breeding ponds, a rearing pond for hatchlings, floating islands, solar-powered freshwater, perimeter fence, and all the infrastructure necessary for this facility. In fact, it's such a lovely place that we get break-ins frequently, whether that's Marlin's crocodiles or in this case, at Trichemis. So it's really become a wildlife hotspot. The breeding stock for the center was acquired primarily through confiscations provided by the Belize Fisheries Department, primarily over the years 2013 and 2014. So we currently have about 45 adults. A few of them are sub-adults, and the sex ratio is relatively even, a little bit more females to males. Dermatemys is considered a strictly herbivorous species. And so at our remote location, we have developed a system for providing a natural diet by both harvesting wild plants and fruits, as well as propagating some of their diet, in particular grasses. So they're fed a combination of different vegetation, mostly of the genus Inga and Ficus, Cecropia, and then the passbone grass. We also feed them a variety of other herbaceous materials. At the center, we've had some pretty good success with nesting over the years, which I'll get into. What's so interesting is that we've had to develop the nesting areas in such a way that these turtles would exhibit natural nesting behavior. Because they lay their eggs under the surface of the water in flooded cavities in muck and mud, it's been an interesting challenge to create that for them, but we've done it pretty successfully. So over the last 6 nesting seasons, you can see here on the left the numbers of clutches of eggs per year, from 1 up to this year, 2020, well, end of 19 and 2020, we had 19 clutches. And in total, we've had about 693 eggs deposited. We believe about 20 of these females are reproducing every year at this point. And we've had very good pretty high fertility rate. Every so often we get a clutch without any fertility at all, but usually it's very high fertility, and our hatch success rate is also we're very happy with. In 2020, of the 208 eggs, 190 were fertile and 184 of them hatched. So we're doing pretty well in that regard. In addition to the work at the center, which is all part of sort of a fundamental conservation action, we also conduct a lot of other activities associated with the species. So it's not just a breeding center in terms of what we do. One of them is biannual health checks, which we've been doing for 5-6 years now, where we collect all of the turtles at the center. Every single one usually takes about 3 days. And we work with both vets that come internationally as well as local veterinarian. And this has been tremendously important. We take ultrasound images of all the females and sometimes even the males to look at the fertility and the status of their reproductive state, and use X-ray machines and take all the metamorphic data that you would assume one would take to, and this has allowed us to not only keep track of the health and deal with any potential disease issues that may crop up, but it's also allowed us over time with both these individuals as well as the hatchlings to determine age at sexual maturity. So this is a very important part of what we do every year. We also are very active in an outreach and education campaign that's been going on for the last 3 or 4 years. We helped establish October as National Hicatee Awareness Month, and October 17th is National Hicatee Awareness Day. We worked with tons of schools and thousands of students throughout the country, and lots of local partners, which has been really critical to our outreach and education campaigns. We also, along with TSA, developed a short documentary film, Hope for Belize's Hicatee, which launched the educational campaign 3 years ago and is still played regularly. This is a very important part of what we do. In terms of moving towards the reintroduction of rewild activities, last year was a transformational year because in 2019, largely because of our constant advocacy in our outreach and educational programming and working with partners both locally and internationally, the government of Belize reached out to ask if we would be interested in doing a survey in a particular part of the country to determine, try to get a population estimate, which really there are no adequate, well-conceived population estimate studies so far on the species. So the work last year was fundamental. We were able to acquire a lot of field equipment through this funding, but also equally as importantly, we identified partners to help with these efforts. So Dr. Day Ligon, Denise Thompson, Don McKnight, Ethan Hollander from MSU's Turtle Lab came down for a period of time and worked with our staff at the Hicatee Conservation Research Center, Jaron and Tom. We worked with local partners, and we were able to conduct some field work. The important part of that field work also allowed us to identify release sites. And before that, we really didn't have a clear indication of where possible useful release sites could take place. So this year, we began our first rewilding efforts. This is a collaboration between BFREE, the Turtle Survival Alliance, Government of Belize, Belize Wildlife Referral Clinic, providing the veterinary assistance, and Day and Denise and his team at an organization called Beetle, the Belize Turtle Ecology Lab, which is now a newly formed turtle conservation organization within Belize. So we all determined to conduct 2 experiments concurrently. One looking at early life stage growth, a comparison of putting hatchlings in different treatments, and then later, 6 weeks later, and possibly at another time, recapturing them all in these different treatments and looking at survivorship, growth, health, etc. And at the same time, using those same animals as part of a long-term mark release recapture effort, because most of these turtles are released in areas that we are all now working in the field, led primarily by Day, Denise, and the beetle team. So all of the turtles were weighed and measured on the day of release. We're talking about 100 and maybe I think I have the number in a minute, but a lot of turtles. They were all notched and pit tagged, and they were released in these different treatments. The first location is a closed system. During flooding events, it probably does not connect to other aquatic systems. And here we released 92 hatchlings. This is a site that we regularly research. Another site was a closed system where the species is not present, but does have other species of turtles, and we think has all the attributes of good Hicatee habitat. We thought we'd release some animals there just to look at the feasibility of possibly starting populations in new areas, because currently where Hicatee are found, they're under such intense harvesting and threat that some of these areas may not be suitable for reintroductions or suitable for long-term survivorship of reintroduced animals. So this is part of our experiment. The third site was an open system within a wildlife sanctuary where there's a number of communities present. And just a couple weeks ago, we released 12 hatchlings there. I really enjoyed Emily Wilder's talk a little while ago about their education work and their reintroduction work, working with local schools. We did integrate with the local kids from the community at this location and got community members involved, and each of those single hatchlings were released by, there happened to be 12 kids and 12 turtles, so it worked out perfectly. And in the bottom right-hand picture, you can see a Belize government fisheries official in the green shirt, also helping to release a turtle. The fisheries department were tremendous partners in these efforts, and even came during the time of COVID to this remote village to assist. The two other locations where hatchlings were also released are part of sort of this early life stage growth experiment. 20 were released in our rearing pond at BFREE as a semi-wild habitat. Obviously it's captive, but it's relatively natural. And then 20 were also kept back in our lab and are going to be raised indoors to look at their comparative growth and health over time. All of these actions and many more have played an important role in our efforts, and certainly in the rewilding parts. But I would just want to emphasize probably our most important conservation actions, which I have not mentioned much, which are actually things like investing and identifying and investing in people. The staff here, the interns that have worked here, are critical to the work that we've done. And building and investing in relationships and partners, both local and international, are key to any of these activities. Our unrelenting advocacy, I think, played a really important role in having the government come to us to ask us to help with Hicatee research and conservation last year, which was critical to leading to this first rewilding efforts. And giving recognition to key stakeholders and partners is super important. So in a nutshell, investing in people and getting people invested has been a continuous theme throughout all the slides I just laid out and everything we do. So what's next? We're working on developing a integrated conservation management and recovery plan for the species, along with lots of other key stakeholders. Unfortunately, that has been put on hold because of COVID, but we hope to get back to it very soon. And throughout that, of course, many activities would take place. We're going to continue our Head Start and Release rewilding programs. This is just the very, very beginning. It's all happened in the last couple of weeks. We're going to continue to advocate for the establishment of protected areas, specifically for Dermatemys. We're advocating for the species to become the national reptile of Belize, and we're also going to be advocating for some revised regulations that can help reduce adult mortality and keep the species in healthy breeding populations for many years to come. So this is Thomas Popp. He's one of the most important people throughout this project when I talked about investing in people. None of these activities would have been as successful without Tom. He's the manager at the HCRC. And I just want to finally make some acknowledgements before I take any questions. Turtle Survival Alliance has been a critical partner over the last 10 years, both as a funder and for technical and moral support. Belize Fisheries Department's been key and critical as part of our programming. I also want to mention some of the funders that have made our rewilding efforts possible. Shout out to Zoo New England for providing critical field equipment and supplies. Oklahoma City Zoo, also critical field equipment and supplies. Jacksonville Zoo and Gardens, they continue to support our health checks and provided veterinary support and other types of support. Santa Fe Teaching Zoo College for providing funding for our outreach and education programs, and others. And before I just take any last questions, I also just want to thank Dr. Isabel from the Belize Wildlife Referral Clinic for providing excellent in-country veterinary support, as well as Dr. Shane Boylan and Dr. Elliot Jacobson and others for providing continuous veterinary support and funding their own ways down here and just being incredibly supportive. I also want to shout out a special shout out to Day Ligon and Denise Thompson and Donnie McKnight for partnering with us in country for our release efforts and becoming tremendous partners. And last but not least, to Heather Barrett and Tyler Sandville for spearheading our educational program so successfully over these years. And finally, I just would like to recommend to everyone to follow what's on this slide and donate to the Turtle Survival Alliance. I know, like so many organizations, they're having a tough time because of COVID, and so I encourage you all to donate to them today. Okay, so I'm just going to look at chats here real quick, if I can find them. Oh, crap.
Jacob, your first question is, how do you spot the nests when you're looking for the nests?
Thanks, David. I think I need some help. That's Tom Pop's incredible observational skills. The turtles at the center tend to come up during the night, and it's just, don't forget, this is usually during rainy season, so it's raining all the time. But they do test holes. They'll just dig a little small hole right on the edge of the bank, or even in the water. So Tom looks for sometimes little claw prints if he's lucky enough to catch that, but he's also just feeling underneath the banks, along the edges. And we also know roughly the period of time, but we've had nests deposited as early as October and as late as April. So it's a pretty wide window. But it's both observation and literally manually feeling for nest cavities under the water in the mud banks.
Second question is, how many hours do these turtles spend eating, being herbivores? Do you have any idea?
No, that's a great question. We haven't done any studies to look at sort of percentage of the day, but they remind me of a cross between a fish and a cow. They're basically grazing. Pretty much, I don't know about all this, I can't say all the time, but they're grazers. They're constantly nibbling on things and grazing. Great question. I don't have the answer.
To follow up with, how or why do you think these aquatic species became totally herbivorous?
Another great question. There's been some interesting theories about the dinosaur extinction era. When the dinosaurs, the likely cause of the extinction of dinosaurs, where this family of turtles was around. There's been some theories that with the meteor and the massive explosion that took place and the loss of sunlight and the decaying vegetation and the loss of species, that this species of turtle, or its closest living relative, its ancestor, did quite well in that environment and probably just ate tons of vegetation, aquatic vegetation and rotting vegetation. It didn't need the sunlight. It doesn't bask. Maybe during that period of time, over hundreds of thousands of years, or I'm not an evolutionary biologist, so I can't say, but maybe during that period that could have had an influence. But it's an interesting question. I would, the diet studies that have been done by Paul Moll and John Polisar, and possibly others, there are some hints of non-herbaceous material in their diet, but it's like little snail shells or bits of insects, which we all, I think, believe that are probably just bycatch while they're eating these this vegetation. But it's another great question. I wish I could provide more.
All right, the next one is, do you think that underwater diapause is obligatory, or can you collect and incubate those eggs immediately? And are the hatchlings sexually dimorphic, or do those differences develop over time?
Great question. So we've done quite a bit of experimentation with incubation. We haven't had the ability to keep, we almost always collect the eggs as soon as we find them. And we put them in the lab in incubation containers. And keep them in moist, but relatively dry substrate, just like incubating most reptile eggs. Because the natural temperatures of Belize cool down during the winter months, November, December, January, the eggs also cool down. And during those periods, they're definitely going through an embryonic diapause. And they really don't start to develop until the temperatures warm up, sometimes as late as February or March. And then they usually hatch out in June. So we haven't been able to collect eggs and put them at, let's say, I don't know, 29C from day one. We've tried to do that a few times, but not that much success because of power limitations here, because we're totally off the grid with solar. But I don't know. It's a great question. What I can tell you is we've done some experiments where we've taken a portion of a clutch of eggs and submerged them in water. One time, I think 8 feet in a well for 3 months. And after removing those eggs, they were still viable. We've done it for 15 days, 30 days, 45 days. Also submerged in shallow amounts of water. And they've all, or most of them have been viable and hatched and are perfectly healthy. So I don't know if it's obligatory. It's a great question. Another experiment that needs to happen. In terms of sexual dimorphism, no. At birth, they all look the same. They don't become dimorphic in terms of color or anything else until they reach adult maturity.
And for our last question, do you guys have a volunteer program?
We do. In fact, volunteers have played a super important part of all of the work we do, including the work with Hicatee. So yeah, you can go to our website, bfreebz.org, and you can contact us and see. In fact, I think it's out there on Facebook. Right now, good luck because, airports closed. We've been locked down here since January. But one day the world will become a better place, and we'd love to have people volunteer. So please feel free to contact us.
Fantastic. Thank you, Jacob. It's always a pleasure to hear about this enigmatic species.